Bacteriophage therapy is simply using these viruses as antibacterial agents to treat bacterial infections that people may suffer from.
Today, I'll be speaking with Dr. Sabrina Green, Dr. Saima Aslam, and Dr. Austen Terwilliger, who have come to the area of bacteriophage research and therapy from very different backgrounds. Dr. Aslam will join us a bit later to answer questions around the clinical application of phage therapy, and we'll share more information about each of our experts in the description below so you can learn more about their backgrounds. But first of all, thank you so much for joining us to share your expertise with our community.
Thank you.
Yes, thank you.
Shall we jump straight into the questions?
Sure.
The first is the obvious. What is a bacteriophage?
Well, I'm so lucky I get to introduce this because it's my favorite thing to talk about. Although Austen could probably do this very well as well. So bacteriophage is a virus, but it's a virus of bacteria. So just as we get viruses when make us sick, could potentially kill us, so do bacteria. Bacteria get their own viruses. And so this is something I like to talk about because I like to really emphasize that these specific viruses cannot infect humans. So they're not harmful towards us. They're only harmful towards bacteria.
Right. Do they multiply in the same way that bacteria or other viruses do?
So yes, they just like our human viruses use our human cells to replicate, the viruses that infect bacteria use the bacterial machinery to replicate. And so they take over the replication machinery of the bacteria and actually use it to grow more of themselves inside the bacteria cell. Mm-hmm. And then they lyse out the bacteria to then infect other bacteria. So you could think of this as kind of like it's perpetuating a drug. You know, it's always, as long as the bacteria are around, then the phage should be around replicating. And if the bacteria are gone, then the phage should also be gone because they're not able to replicate on the bacteria.
So when you say lyse out, does this mean just exploding out of the bacteria? Is that how phages kill bacteria?
Essentially, yeah. It's sort of like I think more of an implosion. But yeah, it essentially explodes the bacteria in a way. So it makes an enzyme that creates holes inside the bacteria cell wall. Mm-hmm. And so that the holes are not good for the bacteria. And so the bacteria then kind of collapse. And so the phage are then able to come out and to then infect other bacteria.
Right. And does every single bacterium have a single phage that can attack it, or are there multiple phages that can attack each type of bacterium?
So that's the great thing about phage is the specificity. And of course, Austen, if you want to talk about this, you can, but they're highly specific. So they target receptors on the bacterial surface. And so different bacteria kind of express different receptors on their surface. And so because of that, that makes the phage very specific. But there are phages that can target generalized expressed receptors too as well. So it's just sometimes you have phages that kind of can act more as generalists, and you have phages that can be super duper specific. But it's very rare to actually find a phage that are as generalists as like some antibiotics that can target, you know, multiple different species of bacteria. So even though phage can be sort of broad spectrum, they're usually not as broad spectrum as some antibiotics.
Right. And do phages exist naturally in the human body?
They do. So they are inside of us, they're on our skin, they're in the air, they're in the water, they're everywhere. So we're actually just beginning to find out exactly, you know, where they are. You know, the microbiome field is still growing, but yeah, there's where the bacteria are, usually the phage are. So if there's bacteria in your gut, you can imagine that there is a lot of phage in your gut as well.
Is there always a counterpart phage for every bacterium in the human body?
So that's the saying, is that every bacteria has a phage. But really, if we haven't identified all the bacteria, then how do we
Right.
really know that there is every bacteria has a phage? So we're just kind of predicting that every bacteria should have a phage.
Mm-hmm.
So there are some pathogens that we know for sure, like Clostridium difficile, it's difficult to find phages against, especially lytic phages. So lytic phages just kill the bacteria instead of acting like parasites.
Mm-hmm.
And then there's bacteria out there that we're kind of can't culture. And so because of that, we may not be able to tell like if there is a phage against it.
Mm-hmm.
So it's still kind of, what we do know is that likely the bacteria that are causing disease, we can usually find a phage against it.
Do phages have a role in the human body, like keeping the microbiome in check, or is that not understood yet?
Yeah, I mean, so that's sort of, I forgot how it was described in a presentation that I went to one time, but it's sort of, that's the phage's role. Mm-hmm. Like they're supposed to be kind of the one that controls, they control bacteria. Mm-hmm. Instead of like actually, I think one of the questions was, how come bacteria don't overgrow? How come phages don't overgrow and take mm-hmm. over? It's an ecosystem. So the phages are there, they control the bacteria that are overgrowing. And then once those bacteria are down, then the phages go down. So it's just this constant ecosystem of phages that are kind of coming and going mm-hmm. Within our microbiome.
One of the questions was whether phages have natural predators, but it sounds like they don't need them because they disappear when the bacteria disappear.
Yeah, usually, yeah, but there actually have been some studies that have shown that there are some things that do eat phages. So it's, we're still beginning, like there's still so much that we need to learn about this, but, phages do have some predators, but usually, yeah, what keeps it in check is the fact that there is no host. So mm-hmm. if there's no host, no bacteria, then the phages aren't growing.
So if phages are already in the body and already helping to keep bacteria in check, is it possible to activate those phages rather than introducing new ones for therapy?
Yeah, so this is a very interesting question, and I'd like to to add on to what Sabrina had mentioned about how your microbiome is basically an ecosystem, and there is a balance to everything. Same on if you're thinking of being in Africa with the fields, and there are predators and prey, right? And so your question might be, well, if lions are so good at killing herd animals, why are there still herd animals around, right? Mm-hmm. Everything exists in balance, and it's the same thing with phages and the cells they prey on. They are going to exist as just a balance in your body. If you're looking to treat an infection and clear the prey or the bacterial cells in this case, you're going to have to tip those scales in your favor mm-hmm. to deliver a lot of phage to drop down and then clear those cells out. Another thought I had is maybe this question was asking more about the temperate phages, which we'll talk about some in the next mm-hmm. two sections. These are phages which are capable of infecting and integrating their genomes as part of the host genome, so they can lay inside that cell until they find a time that is favorable to copy themselves and pop back out. Now, you can induce these temperate phages or prophages from mm-hmm. leaving those cells if you're going to induce a stress, like say modern drug treatment will certainly be very stressful to the cell and might induce these phages to pop back out. But of course, when they do so, they aren't necessarily going to be active to seek out and destroy new hosts and keep on multiplying in that manner. No, right? They're still going to stick with their same temperate lifestyle. If they pop back out, they're going to be looking for a new host to be their long-term home, right? They aren't going to want to kill it. They're again just going to infect and make a nice home inside and ride things out there. So I hope that was clear from both ends on why you need a lot of phage to tip that scale in your favor, and why trying to turn on temperate phages in cells is not necessarily a good idea for treatment also.
Yeah, that does make sense. It kind of brings us to phage therapy and what that is. So it'd be great if you can explain that, and also how long humans have been using this as a treatment option.
Yeah, so bacteriophage therapy is simply using these viruses as antibacterial agents to treat any number of bacterial infections that people may suffer from. As mentioned, this fits well with their role as nature's check on bacteria in general. As for how long humans have been involved with and using phage therapy, what we know for sure is that we started doing studies on larger groups of people around 1919, and in the 1920s and 30s. That is certainly when you saw more studies with phage therapy on people. That is what we know. But I think it's kind of cool to think that it is possible humans have unknowingly been using phage longer than what has been known. I like to think of your folklore tales of healing rivers and waters. Might have been due to people unknowingly taking up some phage. We can't say either way there, but I like to think that some of those cool stories might have been due to phage.
It's a good theory. I like it too. And how long have each of you been working in the field of phages?
Well,
sorry.
Yeah. I'll go first. I know that you have a much more experience. So mine is going to be much shorter. I started working on phage end of 2016, start of 2017, and the main focus was helping to build Taylor.
Okay.
I am, not much longer than Austen, but maybe like 8 years. But yeah, it's sort of it was my research project as a graduate student. And so I kind of moved on from that to actually helping patients from there.
Okay. And how do you think phage therapy has changed over the last 100 years that humans have been using it knowingly?
Well, at least for one, the first big thing was that we finally, at first we figured out what we were working with, right? In the 20s and 30s, we had some notion that these were very tiny particles, but we didn't really know what they were until the 1940s, when we were able to put them under a microscope that was powerful enough to see these viruses. So certainly the first step was learning exactly what we were working with, and now in modern times, the more that we learn about the hosts that these phages use, and the different mechanisms that bacteria use to infect and hurt someone, right? We understand that those factors can co-purify with phage, right? So now we of course take great care that that these, say, virulence factors and toxins and antibiotic resistance features don't end up in our phage preps. And I'll hand it over to Sabrina to fill in anything else I've missed.
No, that's probably exactly what I was going to say is, people, they, especially in the Eliava Institute. So that was one of the first institutes in phage therapy. They've been doing this for a century, and they're still actually treating patients now. And so there's a lot of knowledge there. And, they're using some of the same phages they were using like many, many years ago. So I think the phages, they know how they work, they know how to use them. But really what has evolved is the technology and our ability to really make safe preparations of these phages.
Can you explain what the current process of matching a phage to the bacteria identified in a patient is?
Yes, the basis of it is that we want to know whether a phage is virulent against the patient's bacterial strain. And what that means is that virus is going to bind to that bacterial cell, infect it, make copies, and lyse the cell. That is typically what a virulent phage is. And how we test for that is, again, first getting that patient strain to us, and we will build a lawn of those cells. So just a confluent lawn of all those patient cells, and we'll spot small volumes of our phage prep on to that lawn. And if the phage are virulent against the patient strain, you will often see a good clearing where that lawn should be. And we can then titer this factor out, meaning that we're going to take that concentrated phage and we're going to dilute it by a factor of 10, 100, 1,000. And if you do that and you're still able to see very tiny clearings called plaques, we will then say that yes, this phage is definitely virulent against that patient's strain.
So in some ways it's similar to antibiotic susceptibility testing in that it's being applied directly to the bacteria in a lab setting.
Yes.
How long does that take? Are there a lot of steps involved?
There are very few steps involved, and that testing can be done in a matter of one day, really.
Okay. So from the time of collecting a sample from a patient to the time of administering phage therapy, what's that timeline look like?
Well, this is a very complicated question, so I'm not sure how much time you have given to this section here, but I would summarize it in terms of three routes: either starting with not having any phage and having to find them, having phage but they haven't been purified and prepped yet to the levels that would be suitable for treatment, and having phage on the shelf that would be suitable for treatment. So I'll start with the longest timeline and work back from there. So if we get a patient strain and we don't have any phages and we just have to go back to wastewater or some other environmental source to find a new phage, that timeline is going to be at least 6 months, and probably closer to 9 months and a year. Really, it's relatively quick and easy most of the time to find new phage, but it takes a long time to purify them to the level that they would be suitable for treatment, and then to sequence those phage again to ensure that everything is looking good, that these are lytic phage, not temperate phage, and that these preps don't have, again, a lot of host factors, toxin virulence factors, and antibiotic resistant features present. And then you have to formulate that phage cocktail then, and certified it by any number of measures to ensure that it's sterile and that its toxicity levels are low. That is the longest process. What is quicker is that if we have pre-characterized phage that we know are suitable for treatment, that are virulent against that patient strain, but just haven't been purified yet to the levels that are needed, and a cocktail needs to be made. That timeline is probably anywhere from 3 to 6 months, and the shortest timeline, we have high titer purified pre-characterized phage on the shelf that we know are virulent against that patient strain. We can quick make a cocktail with those phage, certify it with those measures I had previously stated, and you go to a review process with the FDA, just depending on whether you apply for an IND or an EIND. Those timelines are different, but there you are looking at maybe 1 month to treatment. So I hope that was clear and not too confusing for everyone. And thank you for allowing me that rather long-winded answer.
Oh, it's good to know these timelines, because it's not like you can just go to the pharmacy, get a phage, and start feeling better tomorrow. It really is a process, and it means that the patient needs to be in a stable state so that the phage is still applicable in a month or 3 months or 6 months. And if that's the case, at that time point where the phage is ready for administration, do you test the patient again to be certain their infection is still the same?
Yeah, great question, and we certainly do if we can, because the only constant in life is change, and especially bacteria infecting people are no different. They are going to adapt and evolve and change with time. And I think as Sabrina had mentioned previously, phage can be very targeted and specific for their host. So if your host is adapting and changing, you essentially have a moving target. So you want a stable clonal population of bacteria that is not changing. It gives you a clear, steady target to hit with phage therapy. So yes, when we are ready to treat the patient, we would like a most recent clinical isolate to be sent to us from the lab, just so we can retest and ensure that it is still susceptible to those phages that we have first chosen for the phage cocktail.
Yeah, that makes sense. And Sabrina, do you have thoughts to add to that process?
No, I mean, Austen is the main person involved with that process. So yeah, it's very extensive process. I do have to say that it not only is it an investment on us, but also on your clinician. So your clinician is willing to do all this, to apply for this IND or EIND. They're invested in it, and to continuously give us samples if we need, and to work with their hospitals and their pharmacies so that they're allowed to do this. So it's really important that we have good communication with those clinicians, and that you have a good, your clinician is invested in getting this phage therapy to you.
Does sound like quite the process. I have a question about the purification. So without that process, is there a danger that a phage might transfer resistance traits to bacteria that are inside the human body where it will be administered?
Yeah, so again, any feature that is part of the bacterial host being used to purify the phage can purify with those phages, right? And you don't want those factors ending up in your prep. So these high titer preps that are made, and this is part of why it takes long to develop a good cocktail for treatment again, because you're doing a lot of work to ensure that those factors are not there, or that risk is kept to a minimum level.
And you mentioned finding phages in wastewater, which is often how we hear about that in the news, that they found a phage in a sewage plant or in a lake or something like that. Why is that the go-to place for new phages?
Well, just like Sabrina had mentioned in the first section, the logic is straightforward. If you want to find a phage, go to where those bugs are. And a lot of modern infections that people suffer from come from enteric and gram negative enteric bacteria. That means that these are in and a part of your gut on a normal day-to-day basis. So if they're in your gut, they're going to leave your gut too. And when they do, it ends up in with sewage and wastewater, and that is why we can pick up those phages there. And it is a really great resource, because you can pull those phages from millions of people, get a vast array of phages from millions of people in Houston and in other cities.
We have had other resources though. So when we first started, we did go, actually we got it from duck and geese and bird feces and dog feces too. So, you can get phages. It's usually feces, but but you can get phages from those sources if you don't have access to sewage. But I would recommend, wearing gloves and doing everything safely, and that you have a BSL-2 lab.
If you do collect the phages like that, there must be just millions of them in anything that you collect. How do you separate them into individual phages in order to test them against the patient bacteria?
So, I can talk, but I guess Austen can talk too. But it's usually, we are diluting these sewage samples or fecal samples, and then adding it to the bacteria on a plate of what's called agar. So it's like a solidified with LB agar usually. So it's like a solidified medium. And so when you incubate these two things together, the next day, usually you'll see clearings or plaques. And so if you can dilute your sewage sample enough so that you can just clearly see plaques, then you can then isolate that one plaque and then plate it again so that you get an actual individual clonal phage. So then that's how we isolate the phages. It's just by diluting the source that we get the phages from.
Yeah, you don't actually separate out the phages when grabbing your sample. You want everything to be there, and then when you're searching for a phage, for a strain, for which you have new phages, you just let nature take over and guide you, right? You have a giant mix of millions and billions of phage, and if there are some that can infect and win, it'll show up as clearings, as she had just said here.
And then the phages that don't have any effect on the bacteria present will just cease to exist.
Yeah, you won't see a clearing. So it's just, maybe they were there, but they didn't, didn't kill the bacteria. So you can't isolate it.
Okay. And then on the patient side, in identifying the bacteria that you want to develop a phage for, what kind of testing do you use on the patient sample?
So there are several ways that clinical labs can do this. All are designed to be specific for that species or the type of bug. So one method is just using different media and chemicals to grow that patient strain in. Certain species are going to use certain types of foods to grow. Others are not. Some chemicals are going to be toxic to some species, and others are not. So based on that pattern of growth in different types of media, you can use that to infer what type of bacteria you are dealing with. There are some more sensitive and specific methods that these labs will use as well. One is just taking some DNA from the patient strain and sequencing their genome. So there you are looking for what sequences of DNA are specific to certain species of bacteria, and in a similar manner, you can also use a machine and protocol called mass spec, where you are looking at what proteins, instead of DNA, right? What proteins are specific to certain species of bacteria, and if you use all of those methods for I don't know what reason, you would certainly have a much clearer picture of what you are dealing with.
Do you need to be able to grow the bacteria in order to test the phage against it, though?
Yes, great question. And as things stand now, yes, we need the patient strain in order to tell us which phage are virulent or not virulent on that patient strain. Our group and others are looking to build models to hopefully one day be able to predict which phage are going to infect which strain. We just aren't there yet. So for the time being, yes, please send us your strain that we can have in hand in lab.
So for people that are generally culture negative, they can't just show up with PCR or NGS results and hope to get a phage matched.
Sadly, no, and I can't offer anything else at this moment, really.
What's the timeline, do you think, until you can have those models or algorithms to predict what phage might be beneficial?
Well, if we do build those models, it wouldn't be for all pairs, right? You wouldn't necessarily be able to match all phages with all species of bacteria, right? What us and other groups are doing is, we are focused on a small set of strains and phages. So if say there is a subset, a clade of a certain species that has features that we know make it more vulnerable and susceptible to phage, I think we could have those models built in the next few years, and I'm sure other groups would also.
Is there anything else that you think we missed in this section of how it all works?
Yeah, I'm just going to maybe rehash over what I think is most important about this process. It really is about matching the phage to the strain and the environment in which it is found, right? All of those things are going to be happening at the same time during treatment. The phage infecting the strain inside of the patient, maybe in blood, urine, mucus, something like that. It is all about how that is matched, and a lot of science really needs to be done there to move the field forward.
Totally agree with Austen that, I do think that in the future, in the near future, these things will be, we'll be able to predict what phage will infect a bacteria. But currently, as it stands, we can't do that.
Mhm.
But how we actually get to that stage really is by developing larger libraries, by getting more data out there, developing more centers around the world that are all isolating phages and sharing the data. So once we all can, work to that, then these other people that could potentially be left behind because they have unculturable bacteria could get help in the future.
In the meantime, while we're waiting for these new models to come about, why is it so difficult to get phage therapy approved by the FDA in the US?
It is certainly a great question, and different countries and systems work in different ways. What I will say is that the FDA historically was built to deal with very defined chemical structures and drugs, right? It's a very finely tuned and a rigid system, right? This is not what phage are. These are living biological agents that will change just as quickly as their host, right? So they don't necessarily fit into this drug mold. What we're probably hoping for and looking to build along with other countries is a quality by design approach to phage treatment. This is similar to what some listeners may know in terms of the flu vaccine or the more modern CAR T cell therapy. Again, that is truly a personalized medicine to treat cancer patients. And both of those are set up as such as, if you define the pipeline by which your medicine or vaccine is made and you stick to it, then any product that comes out that other end can then be used for treatment. We just don't have that system for phage yet, but I'm sure that the people at the FDA are thinking of it and are looking towards moving towards that type of system. They, like us, just aren't sure what standards they want to set yet and what they feel safe with. But once that falls in place, I think that we can have that kind of system.